All right, so I want to welcome everyone back. This is going to be our very first screencast for Chapter 16. And in Chapter 16, what we're going to be doing is we're going to be looking at the molecular basis of inheritance. And so if you think back to what we had done in Chapter 15, we had looked at the chromosomal basis of inheritance. And so what that means is that we had determined that there was actually a very tiny factor called a gene that was found on the chromosome, and that gene was responsible for the information that was being passed on to the next generation. But what we're going to do in 16 is we are going to look at the molecular basis of inheritance, which means that we're going to look at what actually makes up that gene. And so in this case, we're going to be looking at a nucleic acid called DNA. So we're going to look at both the structure and we're also going to look at how DNA is replicated. All right, so as I had said before, back in 15, we had determined that genes definitely are those tiny factors that are located on chromosomes, and that information was actually gathered by a person by the name of T.H. Morgan, and we had mentioned Morgan back in 15. And what we needed to do now is we needed to determine what are those genes made of. So there were actually two different components that scientists had hypothesized that the genes could actually be made of. One of those was DNA, and the other was actually protein. And what you see down here towards the bottom is an example of both. This representing the double helix nature of DNA, so it's something that you're familiar with. Over here, this represents a protein. And actually, scientists in the early 1900s thought that it was probably protein that was making up the genes that were found on the chromosomes, because they figured that in order to be able to pass on so much information to the next generation, that the molecule had to be very complex. So proteins definitely look a lot more complex than the DNA. And so they had hypothesized that protein must have been that factor that was being passed on. But based on a lot of research that was done in the early 1900s, it was determined that, of course, protein was not that factor, that it was actually the DNA that was found within those genes. Now, the first scientist to do some experimentation to determine that DNA was that factor that was being passed on was a person by the name of Frederick Griffith. And Frederick Griffith conducted his experiments in the early 1900s. And what he had done is he actually had used a very special strain of bacteria called Streptococcus pneumoniae. And what he had done is he had worked with two different strains or two different variations of this particular bacteria. One of them was the deadly or harmful pathogenic strain of this bacteria, and the second strain was one that was very similar to the pathogenic, but it was considered harmless to most organisms. Now, if you look over here on the right, this is going to be a good example of the experiment that Griffith had carried out. Now, we need to understand that Griffith did not actually determine that DNA itself was the result or actually the causative agent in his experiment, but that's actually going to come a little bit later. But over here on the right, as I had said, this is going to be a good example of his experiment. If you notice, we have two different types of cells that we're working with. One of them is called the S cell, and the other is called the R cell. Now, to kind of explain what these cells are doing to these mice down here towards the bottom, what you notice is that Griffith had injected his first group of mice with the S cells. Now, the S cells are considered the pathogenic cells, so those are considered the deadly cells. And he had assumed that, of course, if you inject the mice with those particular types of cells, that the mouse was going to die. And of course, that was the result at the end. His second group he injected with the R cells, which were considered the harmless cells. And when he injected those into the mice, of course, the mouse did survive. Now, the third group, what he had done is he actually had heat killed the S cells. So remember, the S cells are actually the pathogenic cells or the deadly cells. When he did that, he hypothesized that after he injected the mice with those particular types of cells, that the mice would probably survive, which they actually did. So the mouse did not die. Now, for the fourth group, what he had done is he actually mixed the heat-killed S cells and the R cells. Now, remember, the R cells are the harmless cells. Now, his expectation was that the mouse should survive. Because if you think about it, these are the S cells, they were heat-killed, and of course the mouse survived. The R cells, the mouse survived, but what he noticed at the end of his experiment was the mouse actually died. Now, when he did some analysis of the cells of this particular type of bacteria that were extracted from this dead mouse, he found that the living R cells had actually changed or been transformed, and you can see that word right over here, to living S cells. So what he had figured is that there had to be something within those heat-killed S cells that had been transferred over to those living R cells that caused them to transform into something different. So that whole idea of transformation basically means that we have a change in the genotype and the phenotype, in other words, what we see, that is due to some sort of assimilation or taking in of foreign material. Now, if you notice at the bottom here, I did put down the word DNA, but at this point, Griffith, as I had said at the beginning, did not know that the transformation factor was actually DNA. All he knew is that there was something there in those heat-killed S cells that had been used to actually change those R cells into the deadly pathogenic S cells. So the next group of scientists that we're going to look at are by the names of Avery, McCarty, and McLeod. And they did their experiments actually back in the, again, around the mid-1900s, so around 1944. And actually they were the ones that determined that the transforming substance that Griffith had discovered back in his experiments was actually DNA. Now, what you see here in this diagram is a really quick representation of what he had done. So what he did is he took the heat-killed virulent, which basically means deadly strain of bacteria, and what he did is he homogenized the material. And again, these were the killed S strain bacteria. And what he wanted to do is he wanted to basically treat the sample with various enzymes. And if you notice right here in this column, each of these different materials ends in ASC, so that's going to indicate a particular type of enzyme. And in each of his samples, what he was doing with these enzymes is he was basically removing different materials from the sample. So we have 5 different materials that he was going to remove individually from each group. The first one dealt with RNA, so he was going to be removing the RNA from this sample, but he was leaving the other 4, and the other 4 would be protein, DNA, lipids, and the carbs down here at the bottom. So if you notice, when he removed the RNA and he added the treated samples to cultures of R strain bacteria, so again very similar to what Griffith had done, the end result was both S and R strains, so the deadly strain was still produced. Now if you notice for the second group, what he did is he left the RNA, he left the DNA, the lipids, and the carbs, but he removed the protein. This was a protease. And what he noticed is, again, he added the treated samples to cultures of R strain bacteria, so those harmless bacteria, and at the very end, he still got both S and R strains. Now, if you notice down here for lipids, if you notice I intentionally skipped this middle one, he went ahead and removed the lipids, kept the other 4, and if you notice off to the right again, we're still getting the S and the R strains. Down here towards the bottom, he removed the carbs, but kept the other 4, still got the S and the R strains. But if you notice when I go back up here to DNase, in other words, to remove the DNA, but to keep the other 4, the only bacteria that we had at the very end was the R strain bacteria. So again, based on the deductive reasoning of these 3 scientists, it was determined that that transforming substance must have been DNA. Now, 2 other scientists also basically gave us some information to sort of verify that the DNA was the transformative agent. So the 2 scientists mentioned here are Alfred Hershey and Martha Chase. And what they did is they showed that DNA is the genetic material of a T2 bacteriophage. And what they did is they did this by doing experiments with radioactive markers. Now, over here on the right, this is an example of the experiment that they had done. So what they had is they basically had 2 different groups. So they had batch 1 and they had batch 2. In batch 1, what they did is they basically marked the viruses, and these viruses are called bacteriophages, and so they prey on bacteria. So in this case, they marked those viruses with a radioactive sulfur isotope. Now when they marked them, what they did is they actually marked the protein code. So a little background on viruses. Viruses are made of primarily 2 different pieces, or 2 different parts. They're primarily nucleic acid, DNA or RNA, and they're primarily proteins, so those 2 main parts. And so in this first batch, what they did is they marked the protein coat of the virus. And so what they wanted to see is they wanted to see actually what was being injected into the bacteria. Now the reason they have to inject something into the bacteria is because viruses can only reproduce inside of living cells. They can't reproduce by themselves because the cell actually does the work to make more viruses. And so towards the end of their experiment, what they found is that when the virus injected the material into the cell to cause it to start making more viruses, the glowing part, in other words, the radioactive sulfur, actually remained on the outside. So that was the protein part. If you look in batch number 2, what they did is they actually basically marked the second batch with a radioactive phosphorus. And in this case, it was actually marking the nucleic acid on the inside of the virus. And so they did the same experiment, looked at the results at the very end, and what they noticed is that now inside of the cell was what was glowing. And so basically remember that that genetic information is what's going to direct the cell to make more viruses. In other words, what they do is they make more virus parts, they put those pieces together, the cell explodes, and all of those viruses are released in the environment. Well, the protein itself was left on the outside, and so the material that was being injected definitely was not protein. Here, they marked the nucleic acid, again, either DNA or RNA, depending on the type of virus, and basically it caused the cells to glow. So in this case, this was another good example of isolating that DNA is the transformative agent that is being used as the genetic material in this particular case and in the examples we had used before. Now, once we had all of this information, we also had some other scientists that helped to contribute to the evidence that DNA is the genetic material in cells. So another man by the name of Erwin Chargaff, and this again is around the mid-1900s, so 1950, he made observations which basically helped him to establish something called Chargaff's rules. And the easiest way to explain this is to look at the table that you see right here. So what he had done is he had taken various different sources for his DNA. In this case from E. coli, wheat, sea urchin, salmon, humans, and an ox. And what he did is he basically analyzed the samples and looked at the amounts of adenine, guanine, cytosine, and thymine. Now the reason he did that is because he knew, and other scientists knew, that this was one of the basic components of DNA. So remember, we're trying to get closer to the structure of this particular type of molecule. Now what he had noticed is that when you look at the adenine of E. coli and you compare it to the other three, it's actually closest to thymine. And when you look at the guanine number here at 26%, it's actually closest to cytosine. Now, as he looked at the different amounts of these 4 different nitrogen bases through the different groups, he noticed that he found the same results for each of the different animals that he had looked at, or plants that he had looked at. So in this case, again, if you look at the wheat, 28.1, and if you look over here for thymine, 27.4, guanine, 21.8, and cytosine 22.7. So he had the same type of results through. So based on this information that he had gathered, he made the determination that it didn't matter what species of plant, animal, bacteria, living organism that you were looking at, it remained consistent that basically you would have the same amounts of A's as you would T's, and you would have the same amount of G's as you would C's. And so in his mind, this gave him evidence basically that there had to be some sort of association between A and T, and there had to be an association between G and C, basically meaning A pairs with T and G pairs with C. So again, this added to the evidence that allowed us to make the understanding that DNA is the genetic material that is being used and passed on from generation to generation. So now what we need to do is, with the information that's been gathered from these other scientists, it's now time to start to think about building a model of DNA. And there were several people that were involved with this. The first 2 we're going to look at are 2 people by the name of Maurice Wilkins and Rosalind Franklin. Now they were famous because what they had done is they had actually taken a picture of DNA, and you can see the picture down here towards the right. Now this picture was taken using a special process called X-ray crystallography. Now this X-ray that had been produced had allowed scientists to deduce basically that DNA was a helical structure, and it also allowed them to help determine the width of DNA and also the spacing of the nitrogen bases. Now the width suggested that the DNA molecule was made up of 2 strands and actually formed the characteristic double helix that most of us are familiar with when we do discuss DNA. Now, taking the information from Wilkins and Franklin, Watson and Crick, who typically tend to be the people that are credited with building that model of DNA, they were able to construct, based on the pictures and based on some of the chemistry that they knew about DNA, a very well thought out, researched model of this molecule. So they had concluded that there were two anti-parallel sugar phosphate backbones with this DNA. And with this backbone, there were nitrogen bases that were paired in the middle of the molecule. At first, Watson and Crick thought the bases paired like with like. In other words, A would pair with A, and of course G would pair with G, C with C. But they determined that such pairings would not result in a uniform width between the two strands because of the difference in the structure of those different nitrogen bases. So instead, what they found was a pairing between a purine with a pyrimidine. Now, when they paired a purine with a pyrimidine, that resulted in a very uniform width, consistent with the X-ray that Wilkins and Franklin had produced using their X-ray crystallography. So if you look down here towards the bottom, if you notice, if you put two purines together, it became too wide. If you put a pyrimidine plus a pyrimidine, again, based on the structures, it became too narrow. But if you paired a purine with a pyrimidine, the width was consistent, as I had said, with the X-ray data from the previous scientist. So Watson and Crick had reasoned that the pairing was more specific, dictated by the base structures. They determined that adenine, as we had said, paired only with thymine, and guanine paired only with cytosine. So the Watson and Crick model basically explained Chargaff's rules. In any organism, the amounts of the A's, the amount of the T's are going to be the same, and as we had said when we talked about Chargaff, the amount of G's and the amount of C's will always be the same. So if you look over here, as we had said, you have a purine basically pairing with a pyrimidine. A pairs with T, G pairs with C. The difference that you do see between these two pairings is basically found when you look at the hydrogen bonds that are being used to connect the two nitrogen bases. When you look at adenine pairing with thymine, you have two hydrogen bonds, but if you look down here, when you pair guanine with cytosine, you actually have three hydrogen bonds. Now, many proteins are going to work together in DNA replication and repair because our cells are always replicating. So if you're going to replicate a cell, you have to replicate that DNA. So Watson and Crick had noted that the specific base pairing suggested a possible copying mechanism for that genetic material. Since the two strands of DNA are considered complementary, each strand is going to act as a template. Now remember, a template is sort of something that we go by to build something else. So this was going to be used to build the new strand in replication. So in DNA replication, the parent molecule is going to unwind, and two new daughter strands are going to be built based on those base pairing rules that we had talked about in the previous screen. Watson and Crick's semi-conservative model of replication predicts that when a double helix replicates, each daughter molecule is going to have one old strand and one newly made strand. So the old strand is going to be derived or conserved from the parent molecule. In other words, we're going to keep half of that DNA. And then, of course, we're going to use new nucleotides to build that new strand. So what you're going to notice over here on the right is this is going to represent this whole semi-conservative model. So if you think about that original parent cell that had that DNA, on the diagram you're going to notice one of the strands is going to be light blue and one of the strands is going to be dark blue. So in the model that is the accepted model, this semi-conservative model, one strand, maybe it's the dark one, came from the parent. The lighter strand was the new strand that was being built. And so we start off with two template strands, and then of course we build a new strand on each of those. So each new strand of DNA has one old one and one new strand, and that's what's being represented right here in the middle. Again, there were some competitive or competing models with that semi-conservative model back when they were trying to figure this out. One of those was the conservative model. So what they had thought here was that we would have a parent cell, and what that parent cell would simply do is it would make a new copy of that double-stranded DNA. And then there was one called the dispersive model. And what that meant basically was that you would take this parent cell or this parental DNA and you would take bits and pieces of it and you would use that to create the new DNA. In other words, each new strand would have bits and pieces of the old and bits and pieces of the new. Now, based on what we had just talked about in the previous screen, there was evidence to support that semi-conservative model. In other words, there were some scientists that actually carried out experiments to make sure that this was the way it was actually happening. When we look up here, we have two scientists by the name of Messelson and Stahl. And they basically did experiments that supported this semi-conservative model that we had just talked about. In other words, the idea that we have one old strand and one new strand. So what they had done is they had labeled the nucleotides of the old strand with a heavy isotope of nitrogen. And any new nucleotides that were in the sample were actually labeled with a lighter isotope. So looking at this experiment, what you're going to find here is this would be the bacteria culture or the medium that contained our DNA. Originally it was labeled with that very heavy nitrogen, so in this case nitrogen 15, and then it was actually transferred to a medium containing the lighter nitrogen 14. Now the results from that sample found that after you take the sample and you put it into a centrifuge, what they did is they basically separated the newly formed DNA based on the density of the DNA. In other words, if you have DNA that is more dense, it's going to sink to the bottom of the test tube. If it's less dense, it's going to remain more towards the top. Now, the first DNA replication that actually occurred actually produced a band of what we call hybrid DNA. In other words, again, you got to look at the density here. It did not float to the top. In other words, remember those new nucleotides that would be used to construct the new DNA, it didn't go to the top, it didn't go to the bottom. Basically, it remained in the middle. Now, if you think about that, remember we had said it's a semi-conservative model. And so in other words, you have one new strand and one old strand, so that would make sense. So you have half of the heavier nitrogen-containing nucleotides or DNA, then you have half of that strand that has the lighter nitrogen. And so if you think about the difference in density, it's not going to be light, it's not going to be heavy, it's actually going to be somewhere in the middle. So the semi-conservative model basically supports that. When you look at the conservative model that we had mentioned, which again was one of those alternative models, in that case, if it was a conservative model in terms of how that DNA was being replicated, you would have a light band and you would have a heavy band, and that didn't occur. Now, down here towards the bottom with the dispersive model, well, you would get a hybrid because you would have bits and pieces of both found in the two new DNA strands. So if you were to take it into a second replication, you would find that you would actually get two bands with the semi-conservative model. You would still have that hybrid band in the middle, but then you got to remember, as those strands separate, you're going to still be producing new nucleotides with that lighter nitrogen. So we would have more of the lighter bands. In other words, we would actually have a sample that contained some lighter DNA. But if you look at the dispersive model, that wouldn't be the case. You would still have that middle of the ground band because, again, remember it has bits and pieces according to this particular model. Now what we're going to do with DNA replication is we're simply going to take a really quick look at it, and then the next video, video number 2 for chapter 16, that's when we're going to go into a lot of the details of replication. So again, really quickly, just a really close look at how this kind of gets started and maybe the general idea of how DNA replication occurs. It basically begins at special sites called origins of replication. So I want you to imagine the DNA strand, and what we're going to do is we're going to imagine that strand being stretched out so it's no longer twisted in that double helix. What's going to happen is those two strands are going to separate, and it's going to open up something called a bubble. In other words, it's called a replication bubble within that strand. Now, if it's a eukaryotic cell, a eukaryotic chromosome may have hundreds or even thousands of those origins of replication, or those bubbles. Replication is going to proceed in both directions from each origin until the entire molecule is eventually copied. Now down here towards the bottom, this is basically just comparing prokaryotic DNA replication. So we have an example of E. coli, and as I had said, this is going to be eukaryotic DNA replication. So we're still getting two cells at the very end, in other words, two copies of that DNA, but you can definitely see a difference between the two. In prokaryotic replication, DNA replication, you get one bubble that's going to be produced, then eventually you're going to get replication that's going to occur, again, going in each direction, and eventually it's going to create that new DNA. And then, of course, we're going to have two new daughter cells being produced with that new DNA. But as we had said, if it's eukaryotic DNA replication, we have hundreds, if not thousands, of those replication bubbles. And again, each bubble is going to have replication occurring on that strand in opposite directions. So you have several areas on that DNA strand where replication is going to be occurring, and eventually they will all meet up at the end of replication. Now, when you talk about DNA replication, you oftentimes will refer to something called a replication fork. And this is a Y-shaped region where the DNA strands are elongating. And down here towards the bottom, this would be considered, right here, the replication fork of this particular example. Now, there's going to be lots of enzymes that are going to be used as we go through the replication process. And this is just going to be a list of a few of those enzymes. Helicase is going to be the enzyme that actually will untwist the double helix at the replication fork. Then we also have a single-strand binding protein that's going to bind to and stabilize the single-stranded DNA until it can be used as a template for replication. So it's going to make sure, and you can see those down here towards the bottom, make sure that that fork stays open. Then we have one called topoisomerase, which is going to correct overwinding ahead of the replication forks by breaking, swiveling, and rejoining DNA strands. Now, DNA polymerase, which is a really super important enzyme because it's going to be the one that's going to be adding the nucleotides, actually is not the one that's going to initiate or begin the synthesis of that polynucleotide. In other words, the synthesis of that new DNA strand. The only thing that polymerase can actually do is add the nucleotides, and it's only going to add the nucleotides to the 3' end of that DNA strand. And we're going to go back over the 3' versus 5' as we make our way through this. So the initial nucleotide strand that's going to sort of get things started for us is going to be called the RNA primer. So an enzyme called primase can actually start the RNA chain. In other words, help to sort of initiate that RNA primer from scratch. And it's going to add RNA molecules or nucleotides one at a time using the parental DNA as that template. Now, the primer is going to be short, in other words, that RNA primer we had just mentioned, and it's going to be only about 5 to 10 nucleotides long. And the 3' end is going to serve as the starting point for the new strand of DNA. So the typical direction that you're going to find replication occurring is going to be from your 3' to 5' end of your strand. Now what you're going to find is, again, as we had said a little bit earlier, DNA is anti-parallel. So you have one strand going one direction and the other side of the strand is actually going the opposite direction. But again, replication itself is always going to go from 3' to 5'. Alright, so that's going to finish up this very first screencast for chapter 16, and as I said, we're going to get into more details about the DNA replication process in the second screencast, so we'll give you a lot more information that will help to support some of the stuff that we were looking at in this first video. Please make sure that you have completed your screencast study guide before you come to class.